It started with a routine call and an unexpected recovery. Investigators reached a reported set of human remains and found something that did not match what they expected to see. The body looked unusually well preserved for its setting, and the visible proportions felt wrong at first glance. That single detail changed everything. At that moment, only two facts held steady. A body existed, and it needed to be secured. Even before any formal work began, the discovery gathered instant interpretations. Some people treated it like an ordinary death made strange by time and environment. Others insisted the traits proved it was not fully human. None of that counted as evidence yet. What mattered was what could be confirmed on the spot. The remains appeared intact, not scattered, and the unusual features were visible without tools. That visibility fueled rumour fast. The first responders faced a simple problem with high stakes. If the body was human, it fell under legal protection and demanded careful handling. If it was not, the claim still required proof, not excitement. So they worked from basics: limit touching, prevent contamination, and keep track of who approached and when. The discovery shifted from a shocking sight to a guarded object. Questions stayed open, but the clock started. Within hours, the approach changed. What had looked like a strange curiosity started to receive the same treatment as any serious forensic or archaeological find. The first task stayed simple, but strict. Record everything before anything moved. Notes logged the exact position, the condition of the remains, and what was touching them. Photographs followed in sequence, wide shots first, then closer, so later reviewers could see what was real and what might have been disturbed. Then the measuring began. Basic tools established length, limb spans, skull dimensions, and any obvious asymmetry, written down in plain numbers that could be checked by anyone. Each observation got tied to a time and a person, because memory could not stand in for a record. Samples and loose fragments did not get pocketed or passed around. They got separated, labelled, and sealed, so the body stayed one documented object, not a collection of guesses. That paperwork mattered as much as the body itself. A chain of custody log tracked every handoff, every container, every change of location, so later tests could not be dismissed as contaminated or staged. Even the packaging followed rules: clean barriers, controlled handling, minimal contact. In that moment, the discovery stopped being a story told by witnesses. It became a case built from steps that could be repeated, audited, and challenged. Once the first measurements were taken, the skull became the starting point. Professionals logged proportions that did not sit comfortably inside the reference tables they used at the time. The cranium read as too tall for its width, and the face looked reduced compared with the back of the head. That mismatch mattered because ordinary variation still followed predictable patterns. Here, the usual anchors, jaw size, cheekbones, forehead slope, did not line up. Then the limbs raised sharper questions. The long bones appeared uneven in a way that did not match known growth disorders in the manuals available then. Segments looked shortened where they expected gradual taper, and joints looked oddly formed rather than simply worn or broken. In typical human remains, even disease left signatures that repeated from case to case. These proportions did not repeat any familiar pattern, and that absence kept the notes cautious but uneasy. Soft tissue and teeth added a final layer of doubt. The skin and connective tissue seemed unusually intact in places, yet lacked the expected texture changes that came with normal decay or mummification. Dentition was recorded as irregular. Spacing, crown shape, and bite alignment failed to match common developmental ranges used in identification work. Teeth usually told a stable story of age and ancestry. Here they produced contradictions, and that forced examiners to treat the body as anatomically atypical, not merely damaged. The first tests aimed at a simple question: was this one body or a clever assembly? X-rays came first because they showed structure without touching anything. What appeared on the films looked continuous. Bone density changed in a natural way from joint to shaft. The borders where pieces would have been joined never appeared. The skeleton behaved like a single organism, not a collage built to fool the eye. CT imaging followed, and it raised the stakes. Slice by slice, the scans traced the inside of the skull, the spine, and the limbs in 3 dimensions. The abnormalities still looked real, but now they had depth and logic. The joints matched their sockets. The growth lines ran the way living bone usually formed. Even the tiny channels where blood vessels once passed seemed present, not carved or glued into place. Then the work moved to the small scale. Under a microscope, thin tissue samples showed organized layers, the kind that came from biology rather than craft materials. Basic chemical assays added another check. Proteins and mineral ratios fit what bone and preserved tissue typically carried, even if the shapes stayed unusual. None of it proved an explanation, but it did something powerful. It made an obvious hoax harder to defend. Once the first test results circulated, the certainty ended and the arguments began. One camp leaned on medicine. A rare developmental disorder could have reshaped bone, teeth, even proportions in ways that looked impossible at first glance. Another group pointed to death itself. Drying, pressure, and time could twist soft tissue, collapse cavities, and exaggerate angles. The body stayed the same. The story around it started splitting in 4 different directions. Others offered a cultural explanation, and it sounded almost too practical. Some communities intentionally modified bodies in life or after death through binding, shaping, or ritual preparation. Under that view, the anomalies looked less like biology and more like tradition. A 4th explanation went further: misidentification. The remains could have been human mixed with animal parts, or several individuals combined by accident in the ground. Each theory claimed it solved the puzzle. Each raised new questions. As specialists argued, a louder version of events spread outside the lab. Reports reduced careful language into absolutes, and local retellings hardened into certainty. The body became proof of something unknown, a non-human species, a secret experiment, an ancient other. The more the sensational narrative travelled, the more it pressured the investigation itself, because every cautious update sounded like a cover-up to believers, and every unanswered detail fed the same expanding legend. The turning point came when the team stopped arguing about what the body looked like and started asking a simpler question: when did this person live? Tiny samples from bone and preserved tissue went to a lab for radiocarbon dating. It did not offer a perfect answer, but it did something more important. It placed the remains inside a real historical window. Suddenly, some stories lost their footing. The dates did not match a modern fraud, and they did not fit an ancient legend either. They clustered in a period with known burial practices, known diseases, and known movement of people through the region. Then stable isotope tests added a second layer. They read chemical traces from food and water locked into the body. The pattern suggested a specific kind of diet and a likely childhood environment. Context tightened. With that timeline and those chemical clues, the list of explanations narrowed fast. A recent medical oddity remained possible, but only if it matched that era's living conditions. A crude hoax became harder to defend because the chemistry and age lined up too neatly across different tissues, and cultural modifications started to make more sense for some features, while others still resisted it. The mystery changed shape, but it did not vanish. By the time the calendar and context looked firmer, one question still controlled everything. What exactly was the tissue? The next step went smaller than X-rays and scans, down to molecules. Researchers tried to pull genetic material from the most protected samples they had. It did not behave like fresh DNA. It came out broken into short fragments, and it carried signals that hinted at handling and age, not a clean modern specimen. So the team widened the net. When DNA was too damaged to read in full, they looked for proteins, tougher molecules that sometimes survived when genes did not. The first runs returned a confusing mix. Some markers matched human biology strongly enough to matter. Others did not line up cleanly, and the profile looked uneven, like more than one source had ended up in the same tube. That was the warning label. Contamination or mixing stayed on the table. Even so, the results narrowed the story. The strongest signals pointed toward human or something extremely close, rather than an unknown animal or an invented composite. But the gaps mattered just as much as the matches. If modern human DNA had entered the sample during recovery, it could have masked what was truly there. If fragments from different tissues had been combined, they could have produced a false hybrid pattern. The testing did not close the case. It forced the next phase to challenge every step. Once the first results circulated, the case left the original team's hands. Independent labs requested the same raw files, the same sample logs, and the exact steps used to collect tissue and extract DNA or proteins. Specialists checked the basics first. Whether the right bone or soft tissue had been sampled, whether labels stayed consistent, and whether handling had exposed the material to modern human cells. Replication did not begin with big claims. It began with paperwork and process. Then the disputes tightened. Some reviewers argued the genetic fragments looked too short and damaged to support strong conclusions, a pattern seen when old material broke down or when chemicals interfered. Others pointed to warning signs of mixed signals, where human DNA from a handler could hide inside an ancient sample. There were fights over controls. Were blank samples truly clean, and were results repeated across separate extractions? One lab matched key markers, another lab failed to. As more groups tried, the story changed from a single test to a pattern. Teams that succeeded reported which steps mattered, like extra cleaning of bone powder, stricter isolation, or sequencing deeper to separate real ancient fragments from noise. Teams that challenged the findings showed how small mistakes could create a false impression of something non-human, especially when reference databases were limited. The conclusion no longer rested on mystery. It rested on whether the methods held up under pressure. By the time the outside reviews finished, the story changed shape. The most careful teams no longer treated the body as something outside biology. They treated it as human remains that had been pushed to the edges of human variation. Several impossible traits started to line up with known mechanisms. Development that went off track, bones that formed under unusual stress, and preservation that exaggerated details people thought were structural. That shift did not mean the case became simple. It meant the best explanation became layered. Some anomalies fit rare disorders that changed skull growth, tooth formation, and limb proportions in predictable ways. Other oddities matched post-mortem distortion, drying, compression, and chemical changes that subtly reshaped cartilage and thin bone. Even the genetic signals, once rechecked, pointed more toward damaged human material and mixed contamination than toward a separate kind of organism. But consensus stopped short of certainty. The remaining disagreements settled around specifics, not fundamentals. Exactly which condition, or combination of conditions, best matched the full set of traits stayed unresolved. The age at death, the extent of environmental alteration, and whether more than one individual had contributed tissue still carried question marks. The accumulated evidence narrowed the possibilities, yet it also exposed how easily a rare body could look like a broken rule. By the time the argument settled, the case no longer rested on first impressions. It rested on repeatable results. The standing view treated the body as human remains that carried a cluster of uncommon traits, shaped by a mix of biology, damage after death, and the way the tissue had been preserved. The headline claims faded because they did not survive the full chain of tests. What stayed were specific anomalies, mapped and measured, with clear limits on what anyone could honestly say. In practical terms, the body ended up under controlled care, logged like evidence. It remained housed in a formal collection, with access tied to permits, ethics review, and documented handling, while high-resolution scans and lab records served as the main working reference. That shift mattered. It meant the object of debate stopped traveling as a mystery and started existing as data that could be checked. Every sample request, every image set, every method note left a trail that others could follow. Modern tools narrowed the gap between suspicion and proof. Non-destructive imaging reduced the need to cut. Genetic work focused on authentication and contamination control, and results moved through open scrutiny instead of private claims. Uncertainties still remained, because biology and preservation both left room for noise. But the confusion from the first day no longer drove the story. Today, the case stood as a documented specimen with a test history, not a rumor with a body attached.